This is Alessia Neri, PhD candidate from Paolo Palma's lab of Bambino Gesù Hospital and Tor Vergata University in Rome. I will give you a brief overview of our work entitled Children with Perinatally Acquired HIV Exhibit Distinct Immune Responses Against 4-CMV Vaccine. In brief, we compare the safety profile and immunological response after 4-CMV vaccine in a cohort of adolescents healthy control and children with perinatal acquired HIV, named as FIV. We analyzed the B phenotype profile, the serobactericidal activity, the distribution of the antigen-specific B cells, and their transcriptomic profile. The administration of CMV vaccination to FIV individuals is a priority to contribute in the reduction of invasive meningococcal disease and gonorrhea cases in this population. At this moment, only few studies have been conducted on MB vaccination in immunocompromised population, and these studies aim to investigate the impact of HIV on B cell responses to vaccination and antigen-specific antibodies production, defining the molecular mechanism and the signature of suboptimal vaccine-induced immunity to better understand the optimal vaccination approach in this population. Neisseria meningitis is responsible for an estimated 1.2 million cases of invasive meningococcal disease. The development of a four-component group M meningococcal vaccine represents a significant advance in prevention of invasive meningococcal disease. Children living with HIV have shown increased susceptibility to bacterial infection. And the strategic importance of 4-CMV immunization in the FIV population has been further fueled by the recent evidence of its ability to confer the cross-protection to Neisseria gonorrhea. Also, the examination of the differences between participants with different timing of heart initiation, less or more than 6 months, have been investigating. The study was conducted on two groups, FIV and healthy control. 2 doses of 4-CMV vaccine were administered on day 1, T0, and after 60 days, T1. Blood samples were collected prior to the first dose administration, T0, 21 days after the second dose, T2, and 1 year later, T3. Human response to 4-CMV vaccination was measured through the standard human serobactericidal assay, HSBA, at 3 different time points. An aliquot of Neisseria meningitis serum group B suspension of bacterial solution was added to serial dilution of serum of a single donor and then plated and on an agar plate. The colonies of each condition were counted and verified. The serum bactericidal titer was reported as reciprocal serum dilution, yielding higher than 50% of killing. The B cell compartment phenotype was evaluated by flow cytometry in the 2 groups at T0 and T2, and vaccine-induced B cell memory response was evaluated at T2. To investigate membrane antigen-specific B cells, a probe targeting the FHBP antigen was used. To investigate the gene expression profile in non-antigen and antigen-specific B cells, were certain and their transcriptomic profile analyzed by FluidM. The effect of age on the baseline HSBA level was confirmed by a positive association between age and HSBA in the entire cohort. At T2, all participants demonstrated a positive responsiveness to the vaccine, with 100% achieving seroprotection. Comparable levels of HSBA were found between time points T2 and T3 between the 2 groups, with an increase at T2 followed by a decay at T3. However, the ability to seroconvert after vaccination, expressed as HSBA fold change between T2 and T4 greater and equal to 4, was significantly lower in FIV compared to healthy control. The majority of non-seroconverters were found in the late-treated group, suggesting a more favorable response upon vaccination in the early treated individuals. Regarding the B-cell phenotype, a distinct B-cell distribution with a significantly higher frequencies of the activated memory and tissue-like memory B cells subset was observed in FIV. Additional analysis further revealed that activated memory B cells were significantly higher in the late treated FIV compared to the early treated FIV. A distinct distribution of antigen-specific B cells was observed in FIV children compared to healthy control. A higher frequency of antigen-specific B cells with activated memory and tissue-like memory characteristic and a reduction in switched memory B cells was found in FIV. The differential expressed genes upregulated in antigen-specific switched memory B cells in healthy control groups were involved in the regulation of antibody production and memory response. In contrast, in FIV seroconverters, demonstrate downregulation of pro-inflammatory and apoptotic genes, confirmed also when comparing antigen-specific switched memory cells between the FIV and healthy control groups of non-seroconverters. And these findings recapitulate the canonical molecular mechanism underlying the antibody-mediated immune response in healthy control, and on the other hand, FIV demonstrated downregulation of pro-inflammatory and apoptotic signature in antigen-specific B cells. In conclusion, in the present work, we report a deep immunophenotype characterization of the humoral, cellular, and transcriptomic for Sima B vaccine-induced response in FIV compared to healthy control. All HIV participants in this study achieved protective levels of MenB titers compared to the healthy control, and 76 seroconverters were reported in the FIV group, with a similar increase 21 days after the second dose of vaccine and maintenance 1 year after vaccination, but with a reduction of HSV-A fold increase compared to the healthy control. Moreover, FIVs show a distinct B-cell distribution with an increased frequencies of atypical B cells and increased frequencies of antigen-specific B cells in the activated and tissue-like memory B cells and a reduction in switched memory B cells. And finally, the ability to downregulate genes involved in immune inflammation discriminates between seroconverters and non-seroconverted in FIV group, which allow HIV seroconverter to reach a good immunogenicity. Conversely, a classical engagement of key genes required for the development of an effective immune responses upon vaccination have been observed in the healthy control groups. And these differences underline a distinct transcriptomic profile between the two groups. For complete details, please refer to the published paper online now in JCI Insight.